We're going to talk about protein synthesis today. We'll look at the 2 steps of protein synthesis and what the final product is. Protein synthesis. So this is how our cells or microbial cells make protein. With protein synthesis, we want to look at a bird's eye view and the 2 main steps that we would see. The first of those steps is called transcription. And in transcription, what we see is we have DNA, that's where we get our code from, that we can have copied into mRNA. Remember, the M stands for messenger. And this messenger RNA will then take part in the second step of protein synthesis, which is translation. So translation is where we see that messenger RNA going to a ribosome. Remember, these have 2 subunits, the large and the small. And this mRNA is going to feed through our ribosome. And then we have a second player that comes in here, which is the TRNAs. And we have multiple ones of these that'll come through. Remember, T stands for transfer. And these transfer mRNAs carry amino acids. And as this mRNA feeds through and the tRNAs are bringing in amino acids, what we get is a final product of these amino acids added together. Once we have a number of them, we form a protein. And that's the end of the protein synthesis in a bird's eye view. Okay, so we're going to start with that first step, which, remember, is transcription. So let's start with our DNA. And if we have our DNA here, let's put in some bases. So at the top, let's put in some of our bases that we would see. And remember, we're always going to have that complementary base on the bottom. Right? So A's go to T, C's go to G. So this is our DNA strand. And we always have one strand that we would call the template strand, and the other strand is the coding strand. And in order to get that mRNA, what happens is that DNA molecule at this segment that would make up the gene is going to separate. And so what we end up getting is we have this top grouping where we have our A, T, C, and our G, G, C. And we're going to forget about the coding strand for now because this is where our mRNA comes in. So there's our strand of DNA, and we're going to make mRNA. Now keep in mind that mRNA is a little bit different than DNA, right? We don't have the T's that we see with DNA. So instead of A binding to T, we have a uracil replacing it. But everything else works the same way. And this is going to be our mRNA. Now, a quick check on this to make sure that it's coming from the coding is this mRNA should match your coding strand, except for U's have replaced all your T's. So U's replace T's. And you can see that if we go to our coding strand, we have a G to G, a C to C, C to C, G to G, A to A, and here we have that replacement of our T with a U. This mRNA now is ready to go be translated. So this will go next through the step of translation. But let's back up just a second to remember where this is occurring. Remember, we have been studying both eukaryotes, which are organisms like yeast and fungi, and we've also been studying prokaryotes, which are organisms like bacteria. So in order to remember where transcription occurs, cross that T, where transcription occurs, we think about where the DNA would be in both of these. So hopefully you remember one of the big differences between eukaryotes and prokaryotes is that the DNA in eukaryotes is in a nucleus. And in prokaryotes, we have our DNA in the cytoplasm. So just knowing where the DNA is located should tell you where transcription is occurring. For eukaryotes, transcription occurs in the nucleus, and transcription in prokaryotes occurs in the cytoplasm. And that's transcription kind of at a closer level. Our second step then is our translation. So in the first step from transcription, we made our mRNA. And now this mRNA is going to go to the ribosomes in the cell. Remember, it has two subunits. We have a bigger and smaller subunit. And our mRNA is actually going to feed through this in the 5' from the 3' to the 5' direction. And this ribosome now has 3 different sites on it. The first site is what is called the A site, which stands for amino acid. The second site on our ribosome is called the P site, and this just stands for peptide site. And then we have a third that is the E site, and this is the exit site. So this mRNA that we had from our transcription, if we're looking at it from the 5' to the 3', we had our UAG and our CCG. These were our 6 bases that we copied from our DNA. At this point, though, we actually break these into groupings of 3. And these groupings are called codons. They're part of that code that is going to get us the protein that we're ultimately going to make. So as this mRNA feeds through our ribosome, it's these codons that are going to drive what protein is made. And the way that it does that is our last player in this process is our tRNAs. And our tRNAs are a really interesting molecule. I make kind of a funny picture of them, but it seems to work. These are RNAs that are folded in a way that they have at the bottom of them a place where they can have an anticodon. So if we look just at our first codon on our mRNA, we could have a tRNA then that is going to have the complementary code, which in this case would be AUC. So the A is to the U is to the A, and the C is to the G. And this particular anticodon would have then an amino acid that corresponds to it. It'll always carry one amino acid, and that amino acid will be for the particular anticodon that we have here. And then I will take another little bit to even look closer on how these tRNAs interact with the ribosome. All right, so we're going to continue on with that second step of protein synthesis, which is that translation. But I want to get a little closer look when we start talking about what's happening at the ribosome. So if we have a little bit bigger picture of our ribosome here, we have the 3 sites. We have that exit site. We have the P or peptide site, and we have the A or amino acid site. And remember, we talked about the fact that our mRNA is going to run through this in the 5', 3', so it's going to come through here towards the left. And we have that other RNA that plays a role, which would be our tRNA. So if we go back to those codons as we were talking about them, remember we had codons that on our mRNA where we had particular base pairings. And that's looking at it from the 5' to 3'. What you're going to see is that these tRNAs will come in, and the first one, our first tRNA, would have the complementary bases to this first code. And then our second tRNA that would enter the ribosome would have one that would be complementary to our next base. So we would have a GGC. And each of these have particular amino acids attached to them. In this case, we have tyrosine, which would be the first one. That's the one that corresponds to AUC on this. And GGC, that would be glycine. So the way that this works is when we get to the ribosome, as this feeds through, our UAG would be underneath the P site, and we'd get this first one would move into the P site, and our second one would move into the amino acid site. With both of these sitting here, what we end up doing is we end up then having our tyrosine would then bond to our glycine here. And once these two have bonded, we have a whole step where this tRNA goes here. Our first one will move to the E site. Our second one, the one that had glycine, will move to the P site. And this will leave the amino acid site open where a new tRNA can come along for whichever base would follow, whichever codon would follow here. Once we've moved to the exit site, these tRNAs will be empty. So if we look at our first one over there that had tyrosine, when it comes off, it's still going to have this on the bottom, but it's going to be empty up here, and it will allow it to go out into the cytoplasm and pick up another tyrosine to then join into this whole process again. And as this happens, what we do is we get this long chain of amino acids that are going to start bonding to each other coming off. And it's this chain of amino acids that when it's complete, when the entire mRNA transcript has gone through, that this will fold and do hydrogen bonds to become a protein. And so that in a nutshell is what happens really at that ribosome with the tRNA, mRNA, and the ribosome. Now keep in mind that we always talk about eukaryotes, right? When we're talking about our fungi or yeast cells or anything that is a eukaryote, and we also talk about our prokaryotes, which include bacteria. And in this case, we want to think about where does translation occur. It occurs at the ribosome, but where are the ribosomes in the cell? In eukaryotes, they are in the cytoplasm. Sometimes they may be attached to the endoplasmic reticulum, but they're still located in the cytoplasm. And the prokaryotes are also in the cytoplasm. So we're going to summarize now. We were going over protein synthesis, or the making of proteins. So things to keep in mind are the different players and what's going on. So remember, we have two main steps. Our first one is transcription. So let's think about where this takes place. First of all, in eukaryotes, this takes place in the nucleus because that's where our DNA is. And in prokaryotes, this is going to take place in the cytoplasm. And then what players are involved when we talk about transcription? DNA, right? That's where our segment of DNA or the gene that is encoded. And we also have our mRNA, right? That's the transcript, the actual message. So looking then at the second step of protein synthesis, we have translation. So let's start with where that occurs in eukaryotes. We said that's at the cytoplasm, and really it's on ribosomes or within ribosomes. And in prokaryotes, it's also in the cytoplasm and on ribosomes. So who are the players involved when we talk about translation? We know mRNA is because it's what's bringing the message. We know that the ribosomes play a big part of it. And then we have our tRNAs, which are what ferry in our amino acids. So as we think about protein synthesis, it's going through transcription and translation, and then you really want to think about what's that final product. So from when that one segment of DNA where the code came from, the ultimate product of that then at the very end is a working protein. And remember, this is going on in all your cells all the time. And it's these main players and steps that you need to know for protein synthesis.